Hello, everybody. We're back. Last formal presentation of day 2. Essentially, once again, what we're trying to do this day is to give you an introduction, essentially at a conceptual level, as to why species are where they are and why species aren't where they're not. And so you've heard some basics of distributional ecology from me at the beginning of the day, and you've heard some comments from Luis Escobar about essentially why disease systems are different and how they're different. So now what I'd like to do is go over with you some more perhaps specifics. And essentially, I'd like to talk with you about disease as an ecology phenomenon or a biogeographic phenomenon. So let's talk about this. Remember, send in your questions because we're going to get to the end of this session pretty quickly, and there's not going to be any questions to answer. Okay, there we go. We are ready for this presentation. So let's talk about ecology and biogeography. Ecology is the study of interactions among organisms and interactions of organisms with their abiotic environments. So essentially, this is organisms in context. Now we can immediately split the field of ecology into two pieces. We could call ecology the study of the requirements and needs of a single species in relation to its surrounding environment. So this is a lot like Grinnell's and Hutchinson's fundamental ecological niche, or Hutchinson's cenopoetic niche. And then we can look at synecology, which is the study of interactions among species. So essentially, how do different species or populations of different species relate to one another? And so ecology immediately decomposes into two different subfields that are roughly akin to the A and B circles in the BAM. Right away, I can forewarn you that odd ecological dimensions are going to be much more tractable, and the synecological dimensions are going to be much more complex if you try to do any work in this field. Now let's go over to biogeography. Biogeography is the study of the geographic dimensions of biological diversity. It focuses on questions like, why are species distributed where they are distributed? And how did this diversity, whatever diversity you're talking about, how did it evolve and how did it spread? And what historical factors either enabled or constrained the distribution of the species or that diversity? So biogeography focuses much more on historical constraint in producing geographic pattern. It essentially places biological diversity in a broader spatial context and a deeper temporal context than ecology frequently does. So I've already suggested this reading to you. Again, it's in the shared folder, and by the way, I did just post the link on the Google Plus page for the course. I just published the link to that folder, and in that folder you have access to all of the presentations and a growing number of readings that we've mentioned along the way. So take a look at this paper. Essentially, it gives a bunch of examples that are designed to illustrate how diseases really do obey biogeographic ideas. So let's go through a few of these just to get you thinking. So let's talk about something that's in the news right now, the filoviruses, Ebola and Marburg. I'm going to show you some very dated maps that I developed 12, 13 years ago. These are essentially ecological niche models for Ebola in green and for Marburg in blue. And you can see kind of a continent-wide sub-Saharan distribution of these two viruses. But notice that I'm showing you this distribution on kind of a view of much of the world. And notice these yellow stars that are scattered around the world. Those yellow stars represent other places where either Ebola species or Marburg has shown up. And in fact, not included in this map are also stars that we could place on the map in the Philippines. So how do we get a set of viruses that probably pretty clearly evolved in Africa, but with these scattered occurrences around the world? Let's think about this in a BAM framework. Essentially, we have our abiotic conditions under which our viruses are maintained. They're not very well understood. We have our biotic conditions, which we imagine overlap partially but not completely with the abiotic. But then this bottom circle is the dispersal reach. And probably 50 years ago or 100 years ago, that dispersal reach was minimal, which is to say we really didn't, in the year 1300, we didn't see filoviruses jumping to America because they really never had any way to get there, or to Europe, or to the Philippines. But in the last half century, with human movements, or with human transport of other mammals, specifically monkeys, filoviruses have found a way to essentially ride along with those mammals, humans or otherwise. So the very name Marburg virus comes from the fact that the very first time that scientists ran into Marburg virus was when Marburg virus was imported into Germany in the 1960s. So if I look at these occurrences in an environmental space, as with before precipitation versus temperature, I can see Ebola and Marburg as these colored points out near the warm end of the spectrum. But then I can see these yellow boxes, which are other places around the world where these viruses have occurred. And so in a modern human-assisted world, the M circle for the filoviruses is actually pretty broad and has taken them onto 3 other continents where Ebola and Marburg don't occur. And so this is just the simplest of applications of biogeographic thinking, of BAM thinking, where we're saying that the dispersal reach of a species, or of a family in this case, is broader than the set of conditions under which that family is able to maintain populations. So this is the very simplest of such applications. Let's go into a more complicated example. Here's a world map of plague. And plague has a very curious history because it has shown episodes of dispersal limitation and then essentially worldwide dispersal and then retraction again. So here's a map. The red areas are areas that we could imagine as an original distribution. We could argue about whether it got to coastal China originally, but let's take a Central Asia distributional area as a reasonable starting point. And you all know of the plague pandemics, where plague got to Europe at a couple of points during history, and caused all sorts of massive mortality. At some points, apparently killing half of the people in Europe. The most spectacular of these plague pandemics is what's called the third pandemic, which began in the 1850s and some people say continues till this day, and other people say continued into the 20th century. But essentially, the third pandemic was a function of global scale human movement, and basically rats on ships and their fleas. We heard a lot about the plague pandemics and plague movements yesterday in the talk by Dave Wagner. Now, on this map, what I've shown you is in yellow squares the different port cities around the world to which plague arrived, and there are actually more than what I've shown here. And then the gray areas are areas, very approximate, but areas where you have plague transmission today. And you can see that most of the places where there's plague transmission today coincide with port cities, but not all of the port cities coincide with places where there is plague transmission today. So there's clearly some complexity in here. And Dave yesterday showed you some very interesting ideas about the phylogeny of plague isolates. I'm going to put this in a biogeography context. So let's look at it in a very complicated BAM diagram. So look at this three-way intersection between the two dashed circles and the orange circle. So it's a very small area. And this is that Central Asian original area that I was talking about. As human movements expanded, first when Europeans came to the Orient either by boat or by overland travel, you got those first pandemics. But let's focus on the third pandemic. You expand human shipping movements globally, and plague expands massively. So look at the bottom circle and the difference between the dashed circle and the solid line circle. That is my diagrammatic representation of the effects of global shipping. Essentially, just like with the example I gave you of Aedes albopictus, this expanded M made a much larger potential distributional area available to plague. And so you saw plague jumping in the third pandemic to Australia, Europe, all across North America, the Pacific, Africa, South America, India, etc. And then in a vastly simplified framework, but just to get you thinking about this in a BAM framework, one of the key public health innovations that was made was the idea of isolating people from rats, especially urban rats. And so sometimes by very draconian means, like burning substandard housing, or by pouring cement into basements, as was done in San Francisco, if you just reduce the degree to which humans and rats are close to one another, plague retracts rather massively. And so that's the difference between the dashed circle and the green circle on the upper right side of this figure. And so into the 20th century, as these public health measures were taken that distanced humans from rats, plague retracted from a bunch of areas. So if we go back to our map, for example, here in New Orleans, plague was there, but as public health measures were taken, plague disappeared and did not get established in the natural world of the southeastern United States. And we could say the same thing of Australia, just like Dave commented yesterday. So this is about as complicated as a BAM diagram allows us to explain. But what I want you to think about is that we can use this heuristic tool to contemplate different processes that affect disease distributions. Now, back to some simpler examples. Look at the brown area. The brown area in this map is the distribution of West Nile virus, very approximately, as of 1998. And essentially what West Nile virus was a zoonosis that had fairly minor effects and was occasionally eruptive into southern Europe and southern Asia. When West Nile virus found a way to expand its M circle, which was probably a mosquito or an infected bird hitchhiking across the Atlantic Ocean, either in an airplane or a ship, all of a sudden much more of the world became available to West Nile virus for colonization. So this is just like the example I gave you earlier with Aedes albopictus. The blue areas are a very, very approximate niche model that I created based on the brown areas. And essentially, all I'm showing you is the broad potential distribution of West Nile virus worldwide. When West Nile virus jumped over the Atlantic Ocean, within 10 years, it fulfilled essentially all of the distributional potential that you see in blue in the Americas. So the only thing that was constraining its distribution, at least towards the Americas, was that ocean barrier, M. Now you might ask, why hasn't it gone farther east, for example, into eastern India and Southeast Asia? And that's a good question. It may be that there is some dispersal barrier, just as happened in the Americas, and that if West Nile virus were to be transported to Australia or Southeast Asia, that it would be perfectly capable of being transmitted. Or it may be that there are interactions with other viruses, for example, Japanese encephalitis. I would say this is an unknown. Hopefully it won't expand its range, because the last thing that the world needs is a new zoonosis in a new region. We have enough of that as it is. But that is either an effect of M or an effect of B. Now another more recent example is chikungunya. Luis yesterday showed you this map of chikungunya, and we could say exactly the same things about its potential distribution and its limitation by M. Chikungunya is pantropical, and clearly until a year or so ago was limited by big oceans and wasn't able to cross either the Atlantic or the Pacific and get to the Americas. And then, I believe in March of this year, it appeared in the Lesser Antilles of the Caribbean, and thanks to movements of people, mostly apparently related to tourism, chikungunya has now spread, and we have local transmission occurring in a dozen countries already. So that's an example just like that of West Nile virus, and just like that of Aedes albopictus, where oceans constitute major barriers. Now here's a very interesting example, yellow fever. Yellow fever is a zoonotic disease. It's maintained in apparently primates in Africa and a set of canopy-dwelling mosquitoes. And long ago, which is to say centuries ago, yellow fever apparently was transported from Africa to the Americas and became established as a new zoonosis in the Americas. And there are a few cases each year now in the Americas. Clearly, the biotic element was not a factor in limiting the possibility of yellow fever expanding into the Americas. Clearly, again, it was an issue of dispersal limitation, M. But I took those areas where yellow fever is endemic now in Africa and the Americas, and I created a very simple niche model to explore, well, where in Asia and the Australo-Papuan region might present similar conditions. And I got the areas that are shown in red. That's very interesting because there's been a lot of shipping between Africa and Asia. You would think that the opportunity for transport of this virus has already been explored, and yet yellow fever has never been seen in Asia, Australia, or Sundaland. So very difficult to know why yellow fever has not gotten established in South Asia, Southeast Asia, and the Australo-Papuan region. It could be distributional dispersal limitation, just like its late arrival in the past couple centuries to the Americas, or it could be a biotic interaction that has limited its ability to establish in Asian ecosystems. This is another one of those questions that remains open. So I think maybe too many of the examples that we have shown you so far in this course have been global. And I apologize for that. We're going to get to more local scale examples in coming days. Certainly, as we go to smaller areas, smaller extents, M probably tends to be less of a constraint on species distributions. And certainly as we go to finer spatial resolution, so grain size in our examination, as that gets finer, perhaps B can become more of a constraint. But all of these same potential constraints persist. And I'll give you just a very simple example. Over the years, with some valued colleagues around the country and around the world, I've worked off and on with monkeypox. And here you can see the distribution across tropical Africa of known cases of monkeypox as of a few years ago. You can see monkeypox is chiefly transmitted in the Congo Basin, and in fact in the Democratic Republic of the Congo. So as part of some modeling exercises, which we'll talk about later in this course, we did some very, very careful work with georeferencing. What I want you to notice, again, we'll talk about the rest of this, this project later, but I want you to notice this particular map, panel B, and I want you to notice this big yellow arrow that I put on top of it. Notice that we have hundreds of monkeypox cases in the Democratic Republic of the Congo. But then notice that just to the west, across the lower Congo River, just to the west of the DRC, we have no cases in the Republic of the Congo or in Gabon. And that to me is very, very interesting. So I went to my colleagues and I said, is this a monitoring artifact? Which is to say, could it be that we're just not hearing about, not receiving reports of cases that actually do occur in the Republic of the Congo? And their comment was that actually the monitoring in the Republic of the Congo is probably more efficient than the monitoring in the Democratic Republic of the Congo to the east, and that they're pretty sure that there are no cases there. Now, on the east side of this phenomenon, so I'm talking now about the eastern third of the DRC, again we see no cases. And I asked my same colleagues about that, and they said, well, given all of the political problems in that region, there probably are cases there, they're just not getting reported. So we have a very interesting situation in the Republic of the Congo and Gabon, where coinciding with a potential biogeographic barrier, we seem to have no transmission of this virus. That's a biogeographic question, and notice that we're not looking at global scales and talking about continents anymore. So my comment is simply that we have multiple scales acting here, and very simply we have a lot going on, and it's not at all clear at which scales and at which extents ecology is the dominant process, ecology or synecology, versus at which scales we have biogeographic factors acting. So when we talk about biogeography and ecology, we're talking about a very complex sorting of very diverse factors that may or may not constrain species distributions. We have the cenopoetic or Grinnellian ecological niche. This is an ecology viewpoint, and that sets out essentially regions that present conditions that work for a species or not. We have distributional limitations, dispersal constraints, which essentially constrain where the species is able to explore and test whether there is potential conditions that are suitable for the species or not. Some of those areas on the other side of the river or on the other side of the ocean are simply not available to the species. And then we have this other set of factors, which is the presence or absence of other species. These are the synecological factors, the interactions. This is all a way of saying that we're not talking about a simple phenomenon when we're talking about the distribution of a species. And then, and we'll talk more about this tomorrow, we have all these modifying factors. Presence of humans or presence at sufficient densities, human modification of disease dynamics by mitigation campaigns or eradication efforts or immunization campaigns. We certainly have the effects of socioeconomic status and human condition. All of this is about things that modify the basic transmission pattern into kind of a realized transmission pattern. So we are not talking about a phenomenon that is at all simple or easy to deal with. So look at that. I'm flat on at 11:30. Took me a half hour to give you that brief commentary. We're going to take a couple of minutes break, and I'm hoping that a couple more of our instructors will be here with me, and we will again field questions. Anything's game, which is to say you can ask questions about yesterday, you can ask questions about today's material, you can ask questions about what we'll be doing in the next couple of days, but do send us your questions. Here's the email address to which to send them, and we'll look forward to some good discussion. So with that, I will sign off for just a moment, and remember to go to the Google Plus page to look for the link to the question and answer session.